Hello, welcome to our poster presentation. My name is Tyler Hohenstein. I'll be presenting our poster titled Targeted Protein Expression QTL Analysis Reveals Genetic Markers of Recurrence in Breast Tumors. I'll try to keep things short to stay under the time limit. If you have any questions, please feel free to reach out in the Q&A section. Thank you. So, for a brief bit of background, understanding the biological mechanisms behind cancer recurrence remains a complex obstacle to improving patient clinical outcomes. Germline variants have been identified to influence immune response in the tumor microenvironment, as well as a variety of other cancer-relevant processes, making them good potential targets for cancer therapies or as potential biomarkers for cancer prognosis. Expression quantitative trait loci analyses have been used in the past to link genotype to transcript abundance and attempt to tease out the relationship between germline variants and changes in the tumor microenvironment. However, they are limited by cellular heterogeneity in the bulk RNA from both cancer and tumor microenvironment cells. To address this, in this study, we used tissue microarrays to quantify protein expression separately in both cancer and stromal compartments, in nuclei, and in cytoplasm from these two cell types across 81 cancer-relevant proteins, enabling a protein QTL analysis, pQTL analysis. We integrated this protein expression data with germline genotype data from an Axiom SNP microarray in order to accomplish this. You'll see here in Table A from Figure 1 of our poster, case counts for a variety of clinical features by early recurrence status, that is recurrence under 5 years. We conducted several statistical tests to assess the significance of these clinical features in association with early recurrence status. Those that we found to be significant, that were not highly multicollinear with other clinical features, were included as covariates in our differential expression analysis in Lima that I will go over later. Now, before going over our differential protein expression analysis results, I'd like to first go over our IHC subtype prediction model. So we determined IHC subtype to have a significant association with recurrent status, but we have low coverage for this clinical feature in our overall sample population. We only had 674 cases with complete IHC biomarker status data, that is statuses for ER, PR, HER2, and Ki67. And of those, we only had 286 with subtype determinations made by a pathologist. So we decided to train a support vector machine using a radial basis function kernel on our protein expression data to predict biomarker statuses for all cases. We used an 80-20 train test split on those 674 cases with biomarker status data and performed a grid search for the best performing C and gamma values for each biomarker. We then used the best performing model for each biomarker to predict biomarker status for all cases. And we tested the accuracy of those predictions against the test set. We then extrapolated those predicted biomarker statuses to an overall subtype call for every case and tested the accuracy of those overall subtype calls against the 286 cases that we had with subtype determinations made by a pathologist. We used these predicted IHC subtypes as a covariate in our differential protein expression analysis. We conducted a differential protein expression analysis in Lima by early recurrent status using significant clinical features as covariates. In this analysis, we identified 14 significant proteins across tumor and stromal cells with p-values less than 0.1 and fold changes greater than 2. You can see these significant proteins displayed in figures 3A and 3C in the volcano plot and heat map. log fold changes and adjusted p-values for these significant proteins are shown in Table B. We conducted a pathway analysis using NCBI's DAVID and identified zinc finger proteins, lipoproteins, hormone receptors, and palmitate pathways to be enriched versus the background of all proteins in our tissue microarrays. For our protein QTL analysis, we conducted a cis-QTL analysis using the matrix eQTL-R package using long-term recurrence as an additive covariate. So assuming cis-QTLs reported here also play a role in modifying long-term recurrence as well as the protein expression that was tested. To go directly to the most interesting result from this analysis, you can see in figure 4b, we've zoomed in on this spike of candidate QTLs from chromosome 1 that are with cis QTLs within 1 million bases of the start and stop codons of S100A8. So the most significant QTLs for this protein fall within a CTCF binding motif, which is a known transcription regulator. Interestingly, the top candidate QTL for S100A8 was also the top candidate QTL for S100A9. I think that there is some support that bolsters the idea that there's something going on here with the regulation of these two proteins. These are preliminary results. We haven't yet included all the significant clinical features that were included in the differential expression analysis, but these genetic modifiers of recurrence may serve as potential biomarkers of recurrence or as potential therapeutic targets. Thank you. I think that's all the time I had.